Okay, hi guys, welcome to Biology Online with Henny van Vieren. Good morning guys, welcome back to term 2, and can you believe here we go again with online learning. I think we all did a great job. The whole of New Zealand can be proud of themselves for what we did during lockdown, um, and especially with things like death rate, we did really, really well. So, welcome back. That's the right time. And without further ado, we actually need to get back into work. So last time we talked about DNA replication, when we started to talk about it, and we actually talked about um, mitosis. But yeah, now we need to go into DNA replication itself. So maybe just a quick recap to refresh all of our minds. There we go. So remember, I was very proud of myself for making this animation. My wife just laughed at me. Haha. Anyways, so with the cell cycle, the cell spends most of its life in G1, which is the growth phase. This is where the cell, after it has divided, is now half the size that it was previously, so it needs to grow back to full size. As it's growing, eventually it gets so large that the surface area to volume ratio becomes a problem. And this is when it gets ready to divide. And as you can see here, we are now in what is called S phase. And during S phase over here is when the cell starts to duplicate its DNA or replicate its DNA. Because when you are going to have two identical cells, it needs a identical copy of the previous DNA, one for each cell. Okay, so this is way before it divides, and you can see then we move into, so there we go. We are now a old grumpy old cell, which is basically surface area to volume ratio. So he's too fat and he needs to start dividing, so he's going to create the replication of his DNA, and then during G2 he's further preparing for mitosis, and maybe it's a she, but I think he's got a beard, so let's call him a he. During G2 it will now also replicate its organelles, make sure that the cell that is going to be formed, the two cells, are going to be viable cells. Okay, and then we enter mitosis. So this is basically a quick growth 1, DNA replication in S phase. Why they didn't call it replication phase like R phase, I do not know. Maybe that S is for synthesis. And then G2 is further preparation. So growth 1, growth 2, DNA replication, and mitosis. Okay, so I could put the link down in the description for you, just for the Amoeba Sisters mitosis video. It is really good. Then I want to just remind you the structure of DNA. DNA is deoxyribonucleic acid. Okay, deoxy means one oxygen less. Deoxy, it's been deoxygenated. Ribo comes from the word ribose, which is a sugar. Nucleic acid, and the nucleic refers to our nucleotides. So when you look at the backbone, that's the yellowy, orangey, reddy backbone here. That is a phosphate sugar backbone, and then your nucleic acids are the ones that make the rungs of this ladder. Okay, these two strands, so the one and then the other, because remember this is a double helix, are complementary to each other. Complementary does not mean they are the same. It means that they are complementary, and as we said last time, A always connects with T, and T always connects with A, and C always connects with G, and G always connects with C. So that is the complementary base pairing rule. Very important. You will have to be able to write that down. Okay, so the way that we package it, because if you take all of the DNA in every one of your trillion cells, every one of them have got DNA, and if we put them end to end, it's going to be between 6 and 7 foot long. That is taller than I am. Okay, that's 1.8, 1.9 meters. That is a very long piece of instructions just in a DNA molecule. So to get that into a cell, which you can't even see with the naked eye, is a very tricky process, and with that we actually condense them. Now, let me just jump ahead. You can see here's our ribose phosphate or sugar phosphate backbone, and then with our nucleotides. Now, the way that I remember this is with, we talked about at the Gold Coast. Okay, oh, come on. We talked about at the Gold Coast when we have straight lines for an A and straight lines for a T. Let's see if it allows me to draw here. There's straight lines for my, why does it do that? Straight lines for my A and straight lines for my T. Go away. So A, all straight lines. T, you can see it sits in the middle there. G and C, they both have huge curves to them. Okay, so at the Gold Coast, if you ever go to the Gold Coast. All right, so you can see we wind them. How do we wind them? We actually, it is twisted and twisted and twisted and twisted and twisted until it becomes a lot shorter and a lot more condensed. Then we spin them around a histone, which is almost like a yo-yo, and we keep on rolling them up. Now we have a coil of histones, and we keep on coiling them up and more and more and more, and eventually you have a chromosome, and that is inside your nucleus of the cell. Okay, that is the way we package our DNA. Now, talking about chromosomes. Now, when you're in G1 phase, the normal growth phase, this is how your chromosome pairs look. This one would have been from the mom, and this one would have been from the dad, or it could have been the other way around, doesn't really matter. But they are homologous pairs. It means that if this black line here was a gene, that gene could have been the gene for, I don't know, eye color. Okay. Now, I don't know if your mom and dad have the same eye color, but you would have received one copy of the mom's recipe or gene for eye color, and one copy of your dad's recipe for eye color. And they are the same genes, but they are different alleles of that gene. If the one, for instance, is blue and the other one is brown, or hazel and green, and so forth. Okay, so you can think of it as your mom's cookbook and your dad's cookbook. They can both make lasagna, but they use different recipes. And those different recipes are the different alleles for the same gene, and in this case, the lasagna is the gene. I hope that makes sense. Okay, then we'll call them homologous pairs, and there's 23 of them. Okay, 23 pairs. There's 46 individual chromosomes, and in this case it's called a diploid cell. D for double, which means we have the pairs. Okay, D for double means pairs. Okay, and you can see here that in this case it is a male because they have the Y chromosome. If this was a female, it would have been XX, but males are XY. Okay, now when we go through S phase, where we have to prepare ourselves for replication, or mitosis actually, the cell replicates its DNA. So this number one, pair number one, then makes a duplicate of itself and a duplicate of the other one, and we call these chromatids. But your chromosome is still that one thing because it's connected right in the middle there of the X structure, which is something called a centromere. So we have the duplicates, and they are called chromatids. But can you see, it's still one chromosome, you just have two chromatids. There's another chromosome with the two chromatids. Again, same thing, and these are still identical. The chromatids are identical, and then the homologous pairs are still in there. Okay.
Cells like these prokaryotic E. coli cells
Okay, E.
replicate
coli.
themselves quickly and efficiently.
That is
Part of the process
The
of
cells that we
asexual
actually find in
reproduction
our
is the ability
gut
of cells
bacteria
to make
frequently, and also the
identical
ones we look
copies of
at
their
in water,
DNA
to make sure
before
that there
cell
is no
division
fecal
occurs.
contamination. They grow at like
Prokaryotic cells
duplicate
that
between 20
reproduce
and
by
45
binary
minutes.
fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of two strands of nucleotides which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine, and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semiconservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand.
Just check you have the old strand and a new
DNA
strand in all cases. And that old strand and new strand is what we call semi-conservative. So it's like conservation. It means looking after something, or conserving it, so saving it. And in this case, you had one parent strain and one daughter strain. Okay, so each copy gets half of the original. That's what semi-conservative means. All right, and here's another one, DNA replication. So what's going to happen is, now you do not have to know the names of the enzymes, but you do need to know that it is an enzyme doing it. So the names are not that difficult, okay? But as I say, you do not need to know them. So please don't feel overwhelmed by the names. Now you can see the phosphate sugar backbone, and then you can see the nucleotides over here. So along comes an enzyme, and this enzyme will now uncoil it and also separate the 2 strands. Now, for interest's sake, this enzyme's name is helicase because we have a helix, so it's a helicase. And as I mentioned earlier, usually an enzyme will always, well, not always, will usually have ASE, like protease, breaks down protein. Helicase opens up the helix. Okay, so the image just showed there the complementary base pairing, and DNA replication can only happen in one direction, and that is from the 5' to the 3' direction. Okay, so here comes helicase, and helicase is basically unzipping this thing for us. Now we have exposed base pairs. Now you can see this is called the replication fork, but because we have these exposed base pairs, and think back to, remember this is chemistry, this is biochemistry. Now what's happening now is you have these free floating nucleotides in the nucleus, and they need to join up with this, and something needs to help them, an enzyme again, because enzymes are catalysts and they help the reaction to take place, but you need to expose them to be able to add something onto them. Okay, and in this case it is called DNA polymerase. Okay, and DNA polymerase is the guy that reads it, but before that comes the primer. The primer is to prime something, is to prepare it to get it going. Okay, so in the old days, um, when you had to prime a gas stove or something, which means that you had to put in a special field to get it hot and then it will actually run on its own. So a primer is to get it ready. So the primer basically tells DNA polymerase, hey, DNA polymerase, you need to start working from here and you need to work in the 5' to the 3' direction. Okay, so it puts down the primer, and that's a specific sequence that DNA polymerase, the other enzyme, will now recognize and say, oh, here's a start point. Okay, I need to work from here. And here he comes. Now, you can see why the binding site is so specific and why the shape of the enzyme is so specific and so important. And here come the DNA bases or the nucleotides, and they will be, as I say, the DNA strand is going to be created in the 5' to 3'. So 5' moves on and 3' goes the new one. Okay, and in a complementary fashion. Now this is the leading strand. Unfortunately, on the other side, this is called the lagging strand. It cannot happen in that same way because you can only build it 5 to 3, 5 to 3. But if you think about it, where is this coming from? It's coming from this side. I need to go 5 to 3, and then a new piece is unzipped. So here comes primase, puts down my primer. Then comes DNA polymerase and starts building in the 5 to 3 direction. Okay, so building in the 5 to 3. Now a new section is unzipped. And as this new section is exposed, primase has to come again and say, yo, DNA polymerase, can you please start working over here? We have a new exposed piece. And then it comes and closes that up. Now we call these things Okazaki fragments. Endonuclease, again, you don't have to know that, but that's simply taking out the primers, and when the primers are out, DNA polymerase will close that gap, just to make sure that it is completely complementary. And now, ligase comes. Ligase will, well, I can't remember what ligase does, but anyways, don't worry too much about it. Yeah, you have two identical copies, one with the original strand and the other one is a new copy of that. Okay, so that is DNA replication, and you need to be able to tell that story. So again, we have the replication fork, which is right here, and this is where helicase unwinds it. DNA polymerase is building it all the time. Primase, put a primer down for it so that we can start. DNA polymerase knows where to start. It is building in this direction. Let me just turn this into a pointer so that we can see easier. We're building in this direction, which means this guy, this DNA polymerase, has new open exposed nucleotides arriving all the time. Okay, but over here we don't have that because we have to build in that direction. So we're building and closing the gap and then jumping back, and there's a new gap now. Building and closing the gap and then jumping back because now there's a new gap. Building and closing the gap. Okay, and this is called the lagging strand, and we call those Okazaki fragments. Alrighty, so you need to also know that the parent strain, the parent is called the template, and your template where you will then have your complementary base pairs going onto them. Okay, and always remember A to T at the Gold Coast, G to C. The curvy ones go with the curvy ones, and the straight line ones go with the straight line ones. Okay. Now, just for interest sake, we replicate our nucleotides about 3,000 every minute. But E. coli, that bug you looked at earlier, 30,000 per minute. So it grows literally 1,000 times faster than we do. Now, semiconservative we talked about. it's where both of the daughter strands will have a copy of the original strand in there. Okay, so you have one new strand and one original strand, or one parent strand. So this is Drosophila. You can actually see the chromosome. Drosophila is the fruit fly, and a lot of work was done on this early on in DNA research. And you can see, here's your double, that's your replication fork there. There it is split up, and your DNA polymerase will run along, and on this side it will build the Okazaki fragments and constantly replicate this whole thing until it is done. Okay, and let's just zoom in a bit, and you can see there, you can actually see the nucleotides, or the sugar maybe. But you can see that you have your replication forks, and then you can see the two strands over there. Okay, so just remember, you also need to know the structure of this. So phosphate, sugar. Can you see this sugar? The 5 side is pointing up. This one is pointing down. And that 5 actually refers to a carbon molecule that sits right there at that bend. And the 3 side refers to the carbon molecule that sits here at the bottom. That's just what they named them. You don't need to really understand that, but that's why it's called, this is the 5 side, and that bottom one there is the 3 side. So this one goes from 3 to 5, and you can see it's upside down. So if they do ask you to draw it, you have to remember to draw it upside down. And this one is right side up, so 5 prime to 3 prime. And the nucleotides, the sugar, the base, and so forth. Okay, just regarding the history and the what do you call it, the discovery of DNA. Two guys who won the Nobel Prize was Watson and Crick, and ironically they were not biologists, they were physicists. Okay, and this was very recent, talking about in the time of your grandparents or your parents, even in the 1950s. No, your parents probably not, but your grandparents definitely, at the time that this was done my dad was a teenager when they actually only first time visualized and figured out the structure of DNA.
As late as 1951, the chemical structure of DNA remained a tantalizing enigma. At Cambridge University in England, biologist James Watson and physicist Francis Crick had been working to unlock the secrets of the DNA molecule. But they weren't alone. Several other groups of competing scientists were hard at work trying to solve the same puzzle. Some important facts about DNA were already known. For instance, scientists knew that DNA was composed of 4 bases: adenine, cytosine, guanine, and thymine. They also had been able to infer something about its structure with the help of X-ray crystallography. This technique involved passing an X-ray beam through a crystallized DNA molecule and capturing a vague, shadowy image of its internal structure on a photographic plate. Armed with this information, along with their own knowledge of chemical structure, Watson and Crick began building a 3-dimensional model of DNA.
I wanted an arrangement, where I had a big and a small molecule, and somehow you had to form link bonds. Here's A, and here's T, and I wanted this hydrogen to point directly at this nitrogen. So I had something like this. Ooh. So then I went to the pair and wanted this nitrogen to point to this one. I went like this. Whoa.
Today, you can buy a kit and assemble the structure that Watson and Crick put together. But we're getting ahead of ourselves. Watson and Crick needed more data. With the help of physicist Maurice Wilkins, they gained access to an X-ray picture of a DNA molecule that had been taken by his research partner. Her name was Rosalind Franklin. Without her knowledge, Watson and Crick used the data from that X-ray image and successfully completed their model. In a 1953 issue of Nature magazine, Watson and Crick revealed their amazing discovery to the world. Their model showed that the DNA molecule was a double helix. The twin strands were composed of pairs of the 4 known bases linked together by hydrogen bonds. And the whole structure corkscrewed like a spiral ladder, which could easily pull apart in order to make copies of itself with the same encoded genetic information. Watson and Crick had won the race. The discovery of the structure of DNA sparked a scientific revolution. It illuminated the molecular and biochemical foundation of life in a whole new way. It opened doors for areas of research and other great discoveries that few ever imagined possible. As for Watson and Crick, their discovery of the double helix, along with Maurice Wilkins, won them a share of the 1962 Nobel Prize. And what of Wilkins' colleague, Rosalind Franklin? Despite her contribution to the discovery, she wasn't considered for the '62 prize. The rules state that it can only be awarded to a living recipient. Rosalind Franklin died in 1958 of ovarian cancer, most likely caused by exposure to X-rays.
Anyways, yeah, I just want to, special mention again to Rosalind Franklin. You heard that her co-worker gave her photo without her knowledge to these 2 guys who then used that photo to figure out the exact structure. We don't know if any of you remember Dr. Thornton Pett, who used to work at our school. He was working on a lot of X-ray crystallography as well. Not on DNA molecules, but when he looks at that, he can see what it is. But yeah, it's very sad that she was never recognized with the Nobel Prize, because unfortunately she did pass away. But yeah, it feels to me a little bit wrong, a little bit unfair. She needs to have a lot of people know about Watson and Crick, but not everybody knows about Rosalind Franklin. Anyways. Okay, so this is the Amoeba Sisters DNA replication. The link is on the OLE, so can you please go ahead and watch that now. These guys are really good. And that is the end of today's lecture. Anyways, guys, I hope this makes sense. Please also complete the few pages that I gave you in SciPad, just to make sure that you can remember all of this information. Alright, I hope you have a great day. Thank you so much. Bye.